Hello, everyone. This is the session on genomic technologies. We have 6 excellent talks. And I'm Anton Nekrutenko, I'm at Penn State. And without further ado, we will start with Shujun, who is assistant professor in Ohio State. And he will talk about probably one of the very important subjects in the genomic technologies, how to find the repeats. Please.
Hello, everyone. Thank you for joining my talk. Today, I will talk about EDTA2, a de novo transposition annotation in diverse T landscapes. So we know that transposable elements are abundant in eukaryotes. We have lots of huge mega genome projects ongoing right now, including the Earth Bio project, Biogenomics project, and the Darwin Institute of Life, and the 10,000 plant genomes and the VGP projects. So all these projects producing genomes in a daily basis, and the bottleneck come down to how do we can annotate these genomes more effectively. The one challenge of the annotation is the diversity landscape among all these eukaryotes. So here are some examples. Most of them are plants with some animals, ranging from a very small genome size into the human genome of 3 gigabases. And this genome have varying contents of TEs, including plants have lots of LTRs, humans has lots of DNA elements and non-LTR retrotransposons. So the way to annotate transposons in many diverse genomes are 3 ways. First, you base on the copy number, and if the sequence is highly high copy, you think this is a transposons, and homology base and structural base. And among all these, I think structural base is the one that can adapt to the diverse genome landscape, because here is some data showing I compared several genomes that are close to each other or distant to each other, like fly, Madagascar, or erector. You can see they have over 95% genes are shared, but only less than 15% of TEs are shared. And if you move a little bit further away between human and mouse, basically you have no TEs shared. That means homology-based method is not very effective for the diverse genomes. And for this talk, I will talk about the structural-based method. And the structure of TEs are based on their classes, including LTR retrotransposons, lines, and signs for the class 1 retrotransposons. And class 2, we have terminal inverted repeats and helitrons. And previously, I developed a program called Extensive de novo T annotator that can perform annotation for LTRs, TIRs, and helitrons, and with lots of filtering, and eventually produce homogenized, high-quality annotation for the whole genome. And we have lots of users for this method, and because it can deliver very highly consistent annotations compared to other methods. For example, the annotation inconsistency that describe how we name the same similar sequence consistently or not. So the lower the better. So we have a pretty low inconsistency in EDTA compared to other methods, although we still have a pretty high inconsistency for helitrons. And since the publication of EDTA, we have gained over 800 citations. That means people apply EDTA on all sorts of genomes, including many plants and even some animals. And of course, we receive community voice trying to improve EDTA further. The voice is due to the large user base, and including the voice for how to adapt to more diverse TE landscape. And other voice including annotation on non-LTR retrotransposons and how to install the program easier. And of course, we provide the response on how to make it work on many diverse genomes. And one of the challenge is on the non-LTRs, sign and lines, and mostly dominating in animal genomes. So lines has two motifs, but in reality, they have many more complicated structures. And that was not available when we developed our program, but now it's available as named as Endosign. It was developed by researchers in the City University of Hong Kong. And I work with the researchers, and we developed Endosign version 2 that can make the installation much easier and faster, so that and we can also adapt it to animal genomes, so that we can annotate both plant and animal science de novo. And previously, running Endosign on larger genomes was impossible because it was very slow, and now we improve it. And you can see the acceleration is 3 to 9 times faster comparing to the original version. And these are the time that it took to annotate it. And some genomes are too large and too much sign that we can't even execute through the genome. And this include human, mouse, and zebrafish, these model species. But with the improvement, we can now run this program on a relatively small amount of time and produce high quality sign annotations. So this is a benchmark comparing to the sensitivity across similar programs such as SignFinder and Repeat Modeler 2. Among zebrafish, mouse, and human, the 3 model system, we have high quality sign as a gold standard. And you can see the sensitivity of Endosign version 2 is generally better than other genomes. And so now we move on to the improvement on this line annotation. So we observed that the EDTA version 2 had a pretty good sign annotation comparing to EDTA 1. Basically, we had very low sensitivity of sign annotation. And now we incorporate the line version of Repeat Modeler to EDTA 2 that we can improve the annotation in the higher quality. And this is a mouse. And we also test in many other genomes, including rice and zebrafish. And this is the average of all the genomes we benchmarked. And previously we had a very low sensitivity, and now we gain sensitivity for line annotations and reached to a relatively high level, and with also with high specificity. So we add Endosign version 2 and the Repeat Modeler component of line annotation into EDTA, make it the first step of the annotation. And the other part is the TR annotation. Previously, we used TR learner to annotate TRs, but it proves that it's very slow because it has not been updated for many years. So I worked with our undergraduate students here at Ohio State University, and Tianyu is a third-year undergrad that basically rewrite the entire TR learner program and updated its dependencies into the modern level, and including adding a new search engine for searching for TIR annotations. So the updates most focus on reduced read and write on disk, and also add a new search engine and add checkpointing, so that anytime you interrupt it, you can continue your run by picking up the checkpoint. And it's at least 40% faster comparing to the original version of this is original version, and 3 is the latest version. So we modernize TR learner, and then for the annotation part, we observe that repeat masker will usually break degraded TEs into fragments due to alignment extension difficulties for the gaps. And for that, we design an algorithm trying to join those fragmented annotations into a larger piece by recognizing the coordinates. And for that, we can have a less broken annotation more continuously presented to the as a final outcome. And of course, we have many other updates spread out the entire program that we can confidently say this is a newer version of EDTA. And also a collaborator from France, So Juk is trying to update our conda recipe, make it easier. And we also have collaborators from New Zealand to work on the workflow version of EDTA, such as Nextflows and SnakeMix. So this is the overall performance of EDTA. When we benchmark on many different species, including rice, maize, Arabidopsis, those are model plant species, and animal species of chicken, fruit fly, mouse, zebrafinch, human, and zebrafish. So if we average all the benchmark of all these species together, and we look at the sensitivity, and we can see that for the version 1 EDTA, we had very high performance on LTR, TIR, and some helichons because vertebrates do not have active helichons. So basically, they have no helichons in the genome, and that's why we have a low sensitivity here. But you can see the version 2 of EDTA do not maintain the similar high quality on LTR and TIR and also helichon, and boosting the sensitivity for recognizing line and size, and together they count it as non-LTRs. And that's that part is comparable to the repeat modular 2 version for the line and size, but our LTR and TIR and helichon outperformed repeat modular 2. So together, I think EDTA 2 is a better program comparing to EDTA 1. It's improved based on our existing better programs on annotating sign and lines, and also our efforts on improving these programs for their performance, both on the computation and the quality. And also, we extend EDTA from one genome to multiple genomes because we can have dozens of genomes for a single species or for a single genus at the same time, right? So then the idea is to have EDTA annotate on each single genomes independently. Then we can use a pan-EDTA module to screen for high-quality full-length TEs from each of the genomes and construct a pan-TE library. Then we can use this pan-TE library to feed back to EDTA again, so that each of the annotation can have the same set of TEs named consistently. And also we have the false positive filtered through the initial steps. And eventually we have a higher, more consistent annotation, so that allowing us to compare across different genomes. And these are collaborations in different model plant species, including rice, maize, and Solanum genus. This paper is released in bioRxiv recently and has been under review. So in summary, we have a faster and animal adapted annosign version 2 that enables EDTA's sign annotation in animal genomes. And then the accurate line annotation from repeat modular 2 enables EDTA's line annotation. And then we modernize TR Learner 3 and to make EDTA's annotation for TIRs faster and also easier to install. And then we show that EDTA 2 outperforms the original EDTA and also the repeat modular 2 for varying kinds of transposable elements. And with that, I want to thank my group in the Ohio State University, my postdocs and my trainees. And I also thank my collaborators in City University of Hong Kong that work with them to improve annosign. And then my collaborators from Max Planck Institute for Biology of Tübingen, and also collaborators in France and New Zealand to try to improve the installation of EDTA. And with that, I would like to take any questions you may have. Thank you.
Shujun, thank you for this talk. So one of the questions is, and one of the problems with repeat annotation that sometimes it takes longer than assembly itself. So I mean, people who are actually who've done this on large genomes, they know. So in terms of performance, do you see any ways to improving further with EDTA? Because it's still not very fast. Do you have some ideas there?
Yeah, this is a happy question because a couple of years ago, assembly was still much longer than annotation, and now annotation become a bottleneck. And yes, we have a project ahead, which is to compartmentalize different part of EDTA into a workflow strategy so that it can be distributed across multiple servers. This is the work with a group in New Zealand, Planet Food, and is to create a next flow version of EDTA so that it can be faster in a way that it distributes its computation load.
Perhaps we can work together on doing something on the Galaxy side as well, because we can also utilize various resources for different subtasks. So,
yes, of course. We already have a version of EDTA on Galaxy, including the Pan EDTA. That was a work 2 years ago, and then we just need to update it. And so that people can upload the genome and then click through the parameters, and then they can run their jobs through Galaxy.
We want to really run this in systematic way on all high quality VGP genomes. So I think I will be in touch with you on that fairly soon. So, well, thank you. One question here is, maybe we have one, does EDTA require runtime intensive species-specific library generation step comparable to repeat modeler 2? This is the question from Katarina Hauff.
It's difficult to compare because repeat modeler 2 use a different way to classify TEs, and it use coding HMMs to classify, and if that TE do not have a coding region, that you cannot classify, and that's why people see lots of unclassified unknown fragments repeats from repeat modeler outcomes. And this is truly a challenging question, and what we do is to use a structure of the TE and recognize it, and to a structure of the other TEs that we know, and then we name it that way. And the other way can be machine learning, which has been proving quite effective. But we're still trying to find a more accurate way to classify unknown TEs more accurately.
Thank you.
Thank you.
I think we can move to the next talk.
So this talk is about a new method for eukaryotic whole genome structural annotation of protein coding genes. I suggest that users of tools are first looking to the left, and that method developers are first looking to the right panel. For the task at hand, so-called hidden Markov models have been used since 1994, and some of the programs used extensively today have been written about 20 years ago, like my own Augustus. It's commonly believed that genes that are only predicted using the genome as input, so-called ab initio gene predictions, are not reliable. However, many genome annotation pipelines integrate HMMs as core components with extrinsic evidence from RNA-seq and protein alignments to the genome. For example, the NCBI eukaryotic genome annotation pipeline uses the HMM Gnomon, which was designed after the pioneering program GenScan. In our Breaker 3 paper from earlier this year, we have benchmarked it against general purpose pipelines that we could run ourselves under controlled conditions on the available extrinsic input, and found that it performed best. I'm here not considering the application case of annotation mapping between close relatives, where tools are better that fully rely on alignments. A long period of relative stability in methods has been ended by Alessandra Denton and her team, who have shown with Helixer that it's time for deep learning to take over gene finding. Still, HMMs play a central role. The Python code here on the right shows the well-known forward algorithm for a simple HMM. You can take 3 messages from it. The HMM reads a sequence of letters and outputs a vector for each position in the sequence. Just like the transformers you probably hold dearly by now. Modern code can be very short and simple, in contrast to, say, Augustus, which has more than 50,000 lines of C++ code. And you can see here that high-level standard linear algebra functions like matrix multiplication do the main work efficiently and in parallel. So although the introduction of yet another ab initio HMM gene finder may appear to be of little practical relevance, I ask you to bear with me. This sketch here visualizes the layers of our neural network. From a bird's eye view, Tiberius translates a nucleotide sequence at the top here to an equally long sequence of biological labels at the bottom. The output is a GFF file. The layers upstream of the HMM are similar to the ones of Helixer, which served us as an example. The HMM layer computes posterior probabilities of, in this case here, 15 labels during training, and during inference, it computes gene structures. As a test species, we chose the mammal with the arguably best annotated genome, human, as well as 2 others for diversity, cow and beluga whale. For hyperparameter tuning, we used 2 species here in blue, and the remaining 32 species here in black were used for training. And we imposed a lower limit for the phylogenetic proximity between training and test species. So for example, here in human, the minimum time to the most recent common ancestor of a training species to human is 43 million years. And in the case of cow and beluga whale, the closest relative is camel at a distance of 64 million years. Yeah, this is showing here at the top the state transition graph of the hidden Markov model. So this is the matrix A that was in the code on the first slide. It formalizes the inductive bias. Here, the basic biological knowledge about gene structures, that you have splicing there and how the translation works with codons. Our HMM has states for exon boundaries, such as splice sites, these surrounding here the exon states. In our ablation study, it turned out that it's important for the gene structure accuracy to explicitly penalize differences at the boundaries, even if they are small. Our HMM layer is a regular machine learning layer. In particular, the back propagation algorithm of TensorFlow differentiates through the layer so that the full model is learned end-to-end, in contrast to Helixer, where the HMM is just a post-processing step independently written in another language. Another difference to Helixer is that we implemented parallel versions of the forward, backward, and Viterbi algorithm, which reduced the runtime 17-fold on our A100 GPU. We tested Tiberius and these other programs here at the bottom for whole genome annotation of protein coding genes. And here I'm showing the relatively strict accuracy measure on the gene level, where a gene would count as false positive if there is even a small difference. So for comparability between the methods, we chose only the longest isoform of a gene, if there are multiple. So this is both for the reference set, the refseq annotation, and for the evidence-based pipelines like Breaker3 and Breaker2 and Galba, which predict alternative splicing. Tiberius is here the blue triangle. It's even a bit more accurate than breaker 3, which is here the violet pentagon. And this is so even although breaker 3 was given for each species 10 libraries worth of RNA-seq data and protein sequences from AuthorDB. And thereby we excluded the same clades that we had excluded during training of Tiberius. So if you compare apples to apples, that is Tiberius to other ab initio programs, that is Augustus and Helixer, these are here, we see that Tiberius hugely outperforms Helixer and Augustus. It's more than a factor of 4 more, by a factor of more than 4 more accurate than Augustus in terms of the gene level F1 measure. For the human genome, Tiberius was run also in de novo mode, here in the black triangle, where it's used the multiple alignment of 64 unannotated other mammalian genomes. And this pushed the accuracy even a little more and shows that one can get, you can see the number from the paper, 66% of gene structures correct in a complex genome and without doing any RNA sequencing. The gene level F1 of Tiberius, so we think that Tiberius is a worthy successor of Tiberius. We also compiled Busco statistics, which measures the ability to identify the mere presence of certain universally present proteins. The interesting conclusion I draw from these results is that a reasonably good completeness score, such as the one achieved by Helixer here on the 3 species, can go along with a relatively poor gene structure accuracy, as we've seen on the previous slides. Therefore, I do not recommend to interpret a high Busco completeness as evidence for a good gene structure accuracy, and still less as a benchmark criterion between annotation alternatives. Here's an example browser shot with 2 RefSeq genes here shown in red at the top. You can browse the human annotation of Tiberius yourself by following the link here at the bottom. And perhaps as a reminder what errors can be made in the structural annotation, there could be false positive exons, for example, like Helixer does here and here, and split genes like in this range, or even missing genes, like breaker is missing here, the containing genes of this nested pair of genes. Yeah, running time. So the Tiberius average running time for a whole mammalian genome is about 100 minutes here, so just under 2 hours. And in an apples-to-apples comparison with Helixer, which both run on GPU, it's about 5 times faster. And this is probably attributable to our parallel HMM decoding algorithm that we describe in the paper. In an apples to oranges comparison to these tools which run on CPU, we see that it's also much faster than these pipelines here on the right. Although this version of Tiberius was trained only on mammals, we tested how the machine learning model generalizes to out-of-distribution input. So as you expect, the accuracy decline when you increase the distance to the training species. Here we're showing this distance to mouse as an example training species, or as a representative training species. Here are the 3 mammalian test species. This is a chicken, zebrafish, Zoophila melanogaster, and 2 plants. Yeah, so the performance on chicken is somewhat lower than the performance on mammals. Zebrafish performance is clearly lower than what could be achieved with breaker 3, for example. You can see that from the breaker 3 paper. But remarkably, the mammalian version is still much better than guessing on the insect and on the plant species, which suggests the potential, at least, for multitask learning across eukaryotes. it makes little difference whether the repeats have been identified before or not. So we have both versions. We have a version trained on masked genomes that you used on masked genomes, and the version trained on unmasked genomes that we recommend to use on unmasked genomes. So we provide both for download, and it doesn't make a big difference unless you are very distant. Yeah. So here are a couple of example species, like here the plants and Drosophila, where masking helps. But for the species where we would recommend using it, Tiberius et al., it doesn't make a big difference. So the conclusions I draw are that shallow machine learning models like Augustus are finally not competitive anymore. B, that you can annotate a mammalian genome fast and easy with Tiberius. At the moment, an RNA-seq integrating method may even be less accurate than the ab initio Tiberius. So after 30 years of HMMs in gene finding, still only machine learning models with an HMM are competitive and not general purpose language models such as LSTMs and transformers. I here refer to this benchmark paper on arXiv. As future works, we plan, of course, to further improve accuracy using RNA-seq. For this, we will extend the model to predict alternative splice sites as well. We plan to train Tiberius so it becomes broadly applicable on eukaryotes. And we plan to train a genome foundation model, a language model that understands protein coding genes and that can be used also for other purposes, such as predicting transcription factor binding sites. I'd like to acknowledge, of course, the work from my team. This photo shows them in order of the author list on this bioRxiv paper. The preprint at the top. The preprint also holds a lot of the details that I had to omit here on the talk. So Lars did most of the work on Tiberius. Felix did the crucial HMM layer. Katharina compiled the data, and I did the de novo comparative stuff. And you can download the code. You can also train it on your own species and browse the annotations from this link here at the bottom. Thank you for watching.
So this was the talk by Maria Stanke on new gene prediction tool. We have several questions here. Tiberius looks absolutely transformative. This is very exciting. I wonder if you've considered trying to stress test it to organisms that are very phylogenetically divergent from the model species you've shown results from, with unusual traits like G composition extremes or very large genomes. This is the question from Christopher Lemur.
Yeah, and thanks for the question. We haven't yet, so the results are very new yet. So far we don't have benchmarking data on other clades, but are about to test it as well. So we used the human because it's particularly difficult. I mean, at least we have long gene structures, so we would expect it to be on easier genomes to be more accurate, but we'll have to see this. So can't say now.
Are you ready? This is a question from me. Are you ready to start generating Tiberius annotation sort of en masse on large sets of genomes? Because that might be very useful. I mean, most of the default genomes that you find at NCBI, for example, they just have Augustus annotation, which are clearly not good.
Yeah. So, I mean, in terms of training, we'd only have the mammalian, but it's very easy to run it on all mammalian genomes because we don't require any other data than the genome, and so this can run in a loop less than 2 hours per genome. But this can be done by anybody as well. So who has the rights maybe to submit an annotation to GenBank.
What is the current code status? I just looked at the GitHub. Is this sort of ready to be bioconded and
yes. So actually, Felix and Lars are about to provide a conda version for this.
Well,
So
you probably need to use
just
because
try to use to a new TensorFlow version. Your sound was cut off for me.
I mean, you probably want to go the QuantaForge route because of the GPU specific dependencies there.
I
Yeah,
see.
And
Okay, thanks.
And I actually will reach out to you separately on that. This is very exciting talk. I don't see, okay, but last question. Tiberius is very exciting. It is somewhat different from Breaker 3. Can you expand on the differences in terms of input data sets, protein, RNA-seq? Is the larger input better?
Yeah, so it's very different. It's Breaker is a pipeline that integrates all the evidence, and Tiberius is just using the genome. So it's in terms of input completely different. So the current version is easier to use. Let's say if you just have the genome, and it's of course it's the one is deep learning, and a Breaker is using a pipeline where it's basically, it does all sorts of things, alignments and many steps, different gene finding programs. It uses GeneMark, self-training, Augustus, gene finding in transcripts, aligned transcripts. So this has been growing for maybe like 20 something years if you count all the tools that go into Breaker. And Tiberius is very new. We just started developing it last year.
Thank you, Mary. I think we need to move forward, but I'll have some other questions that I'll reach you separately. Oh, okay, so we're continuing. Again, this is Genomics Technologies session, where we have two talks. We're on a third one. The third talk is by Lindsey Hauff from Rutgers, and her talk is on de novo genome assembly of endangered lemur using portable nanopore sequencing in the rural Madagascar.
Hi, my name is Lindsey Hauff, and I'm a PhD candidate from Rutgers University. And today I will be telling you all about our efforts to conduct a de novo genome assembly for an endangered lemur species, Lemur rufifrons, using only portable nanopore sequencing conducted in rural Madagascar. To set the stage where I do my work, Madagascar is a well-recognized biodiversity hotspot, whose flora and fauna are increasingly facing major anthropogenic threats, such as overharvesting and climate change. Notably, there is approximately 90% endemism, including all lemur species, which makes it a prime conservation area and concern for primatology. However, it is anticipated that nearly 44% of forest cover has decreased across the country in the last 7 decades. Many conservationists have been turning to genetic and genomic approaches to try to help identify species or populations of particular concern. However, many of these approaches require the use of a reference genome of either the species of interest or perhaps a closely related species. When conducting genomic work, it's also important to recognize that there are some major global discrepancies in genomic capacity. And this often comes down to the fact that most of our genomic sequencing centers are predominantly located in the global north, as can be seen here depicted in this distribution map by Helmi, Awad, and Maswa from 2016. The distribution of these sequencing centers often result in samples from biodiverse host countries being exported to be sequenced outside of the country. This can provide significant barriers of engagement to genomic research to these researchers of host countries, that is often very costly, and their access to training is quite limited. However, new portable sequencing technology has the potential to try to help mitigate some of these discrepancies. Specifically, our lab uses the Oxford Nanopore Mirian devices that are portable, long-read, real-time sequencers that can be used really anywhere you have access to a stable power source. So now sample collection, DNA extraction, library prep, and sequencing can all be conducted within the host country, and there is no longer a need for sample export. This can help permit in-country genomic capacity building and has a pathway for more equitable international collaborations in genomic research. So our lab took 2 Oxford Nanopore Mirians, the MK1C and the MK1B, which is the one hooked up to that powerful gaming laptop, to conduct whole genome sequencing of Lemur rufifrons or the red-fronted brown lemur, in rural Madagascar. All of our sequencing efforts were conducted at the Central Val Bio Research Station in southeastern Madagascar near Ranmafon National Park. The species that we were sequencing, Eulemur rubifrons, or the red-fronted brown lemur, is a small-bodied arboreal primate that is widely distributed across Madagascar. However, it is threatened by both habitat loss and hunting, and is often considered one of the most commonly hunted lemur species in Madagascar. At the time of doing this work, there was no reference genome available for Eulemur rubifrons, nor was there an annotated genome available for any of the species Eulemur genus. So to do this work, I'm kind of going to briefly overview our methods for the sequencing, including both our sampling, extraction, library prep, and sequencing protocols. This work was done over the course of 2 field seasons. So in 2022, we first sampled skin tissue from an opportunistically collected specimen. So the specimen was found dead in the forest and brought back to the research station for preservation. From this skin tissue, we were able to extract high molecular weight DNA. And as this was our first time conducting work in Madagascar and Central Val Bio Research Station, this pilot study first initially used our field sequencing kit as a way to really test the protocols and determine if this would be a viable process. Additionally, training of numerous Malagasy research technicians and students were conducted during this initial stage. We then returned to Madagascar in 2023 to really ramp up our sequencing efforts to be able to produce enough genomic data to do a de novo genome assembly. So we were able to utilize some of that high molecular weight DNA that was extracted the previous year. And we were also able to extract new DNA, as in between those 2 field seasons, the specimen that we were utilizing had undergone a comprehensive dissection. So now we had access to the internal organs. So we used the spleen tissue at this point to extract more DNA for the project. However, in between those 2 sequencing endeavors, our high molecular weight kit expired, perhaps due to the conditions of the rainforest, and we had to switch our extractions to the DNeasy blood and tissue extraction kits. So using both the high molecular weight and non-high molecular weight DNA, we then conducted sequencing and library prep with a ligation sequencing kit, as we had spoken to many researchers as the yields were significantly higher with these kits, and they were fairly robust to travel. Although they do have to be cold packed, they were able to do so rather easily and get them to the field station without issue. And again, both the sequencing for both 2022 and 2023 were all conducted on those 2 Oxford nanopore devices that are shown below. Once we had all of our sequencing conducted, we then went through the process of generating our pipeline for our nanopore-only reference genome assembly. First, we were able to take those raw reads and re-base call them using a higher accuracy algorithm. Once we were more confident with the reads that we had generated, we then trimmed our adapters using PortChop, and this resulted in over 36.5 million reads being utilized for our genome assembly. And these reads had an N50 of 2600. However, if you think back, we had some of that high molecular weight DNA included as well. So some of those reads were significantly longer than that N50. Using these 36.5 million reads, we then assembled using Fly, which is a long read specific assembler, and polished with Medaka. So our first initial draft assembly had 17,373 contigs. We then went through efforts to clean up and further polish this draft assembly. We first removed potential allelic contigs using purge haplotigs, and then we screened for any possible contaminants and removed those contaminants that were identified using Kraken-2. We then masked repetitive regions of the genome to help aid in downstream analyses. Once we had this complete, we were then able to scaffold our draft genome to the lemur cotta reference genome, which at the time of doing this work was the most complete reference genome available for the family Lemuridae. Now that we had our scaffolded genome, we did some preliminary genome assessments using both QUAST and BUSCO. When we were happy with how our genome was coming along, we then underwent the TOGA pipeline to find initial genome annotation. So TOGA is a tool for orthologous genome annotation which uses a human reference set of about 20,000 genes to try to find orthologs within our new reference genome. Overall, we were very happy with the genome that we were able to produce with our nanopore-only sequencing. Our genome size ended up being 2.16 gigabases large and had, we found, which was near complete and highly contiguous. So there's a lot going on in this snail plot, but I'd first like to direct your attention to the green portion at the top right, which is showing our BUSCO scores. So for our BUSCO scores, we used a primate-specific data set, and from that, it had over 13,780 genes. Of those genes, we found 96.1% of those genes complete within our assembly, and 1.1% were found as duplicates. So we were actually quite happy with the BUSCO completeness that this showed. Additionally, we found that our genome was highly contiguous, which is really indicative of how smooth the snail plot is. Other metrics that are shown here on the snail plot include other general genome assembly metrics, such as our N50, which is over 100 megabases long. Next, we wanted to see how this genome compared to some of those other publicly available annotated reference genomes for lemurs that are an NCBI. So we took our lemur bronze BUSCO scores and then compared them to 3 other lemur species, Microcebus murinus, lemur cauda, and Propithecus cockerelli. And with those 3 genomes, we also calculated their BUSCO scores for that primate-specific data set. And as you can see here from this chart, our lemur bronze assembly is actually highly comparable to those other publicly available genomes. So we were quite happy with how our nanopore-only assembly compared to some of these other genomes that were assembled in the global north. Finally, I'd like to discuss our TOGO results. So our TOGO pipeline was able to classify 19,414 of those human reference orthologs within our lemur bronze genome. Of those 19,414 genes, 79% of them were found to be fully intact within our genome, while we had informative annotations for nearly 96% of those genes. So by informative, it means that perhaps a few of the human transcripts are missing, or whether there could have been uncertain loss about a handful of those transcripts. In conclusion, we were really excited about our efforts and the genome that we were able to produce. We believe we produced a high-quality reference genome for an endangered species and did this work entirely, all of the genomic work entirely within the host country. So not only is this reference genome, we'll hope, will be a valuable conservation genomic resource for lemur bronze as well as the rest of the lemur genus, we were also very excited about how these efforts were able to help facilitate genomic capacity building within Madagascar, as numerous Malagasy collaborators, both researchers and students, had participated in every genomic step of the protocol. I, of course, would like to thank those collaborators that I had just mentioned, as well as the staff at the Central Val Bio Research Station, whose help helped enable this work, as well as the funders that provided the resources to do this work. This paper is currently available as a preprint and is in the review process. So if you are interested, please follow along, and I'd be happy to take any questions now. Thank you.
Okay, Lindsey, thank you for this. So there are some questions. The first one is, what was the approximate time taken by flight to assemble the genome? I had extremely long run times for genomes with higher repeat content. This is Nafia Cheetah question.
Hi, thank you for the question. Flight was not really the limiting step for our assembly process. And if I recall correctly, it was about 24 hours on running on our cluster.
In terms of, let's see, yeah, and okay, another question from Yutang Chen. What's the base level accuracy of the assembly? How about the structural correctness of the final assembly? And there's like 4 questions. Are there any assembly mistakes? And would you consider high-C data for scaffolding? Of course, then you'll have to bring Illumina machine with you.
Yeah, so that wasn't the current scope of this project. Some of the aims of this current objective were to really do the genomic work in country and to use this as a genomic resource for resequencing efforts going forward. It'd be fantastic to have a higher quality reference genome with that high-C data, but just not currently within the limitations of our current study. And also this genome is actually fairly new. We do have other plans of looking into it for accuracy and looking more at the structural correctness. But at the time of doing it, we haven't gotten to that stage of the analysis.
Did you try any other assemblers than Fly?
We were, at the original onset of the project, we were attempting to use a few other assemblers, but just they weren't showing to have the same amount of ease of user access, as well as they weren't showing to have any significant difference of assembly. So we decided, based off what we were seeing with our initial progress, that Fly was going to be our best course moving forward.
This was the question from Anne McCartney, and question from Eric Jarvis. Were you able to calculate QE value, the base accuracy?
We had not done those calculations yet, no.
And purely a logistical question. So how easy it is to prepare good libraries in the field for Oxford Nanopore?
If you're able to have those high molecular weight DNA extractions, is actually quite easy. One of the biggest challenges of doing the nanopore work is just getting the supplies on site. But the supplies that you utilize are some of the kind of basic laboratory supplies. So thankfully, the lab that we were working with was pretty well stocked for those like typical machinery. So once we had all the equipment and things on site, it was actually a pretty straightforward process.
Yeah, because my question was specifically about getting long high molecular weight DNA out of this. So, okay. I don't see any other questions for you. Thank you so much. Okay. So again, this is the Genome Technologies session. We are going to the fourth talk by Luca Degradi, Università degli Studi di Milano. The first two Fusarium verticillioides genomes from human patients, a genomic view. And Luca, please pardon my Italian, but please go ahead.
Good morning, everybody. I'm Luca, and I'm doing my PhD at the University of Milan in the plant pathology lab. The research is funded by the EU within the framework of Next Generation EU initiative. Moving on my presentation is about two Fusarium verticillioides genomes obtained from human patients and possible correlation between biological behavior and genomic information with a special focus on the genomic steps. So, why Fusarium verticillioides is under our spotlight? Fusarium verticillioides is famous because it is able to affect several agricultural products, mostly mice, causing important losses. And moreover, it is able to produce fumonisins, which is a pool of a carcinogenic compound already regulated by International Agency on Research of Cancer. In the recent years, Fusarium verticillioides has also been found associated to human patients, mostly immunocompromised patients, in which is able to cause keratitis, systemic infection, and skin infection. In order to better understand the distribution of the infection of Fusarium verticillioides on humans, I made a research on Scopus database, in which I searched for the papers that reported at least one case of Fusarium verticillioides associated to humans. And in this picture are highlighted the countries in which is reported at least one case. So we can say that also at the clinical level, Fusarium verticillioides is widespread. But no genomic information are available about Fusarium verticillioides that affect humans. So for this reason, we can move on the aims of this work, that is firstly to sequence and assemble the genome of two Fusarium verticillioides isolated from humans, both from blood Italian patients, one from patients affected by leukemia, and the other one immunosuppressed after bone marrow transplantation. And after sequencing, we want to try to correlate biological behavior and genomic information that we obtain after sequencing. So from a biological perspective, we perform several tests, including the growth test at the human body temperature and a fungicide sensitivity test. The goal of this step is to find if there are any differences in behavior between the two isolates from human and the agricultural strains. So for the growth test at the human body temperature, we used the two human strains and a reference, which is the Fusarium verticillioides 7600, which is the most known strain in this of this species. So our results showed that all the strains were able to grow at the human body temperature. Another interesting biological characteristics that has also clinical implication is the sensitivity to fungicides. In order to assess if there are differences toward the azoles that are used, we test 12 different azoles, 4 that are normally used in clinical settings and 8 that are normally used in agriculture. Results highlight that most of the azoles, for most of the azoles, the two human strains present higher resistance, while the only one in which we found higher resistance for the agricultural strain is the prochlorats. Moving on the genomic part, here I report the workflow in which you will find in green the tools used on the Omics Box platform. In orange, you will see the input data. In blue, we have the tool used on Galaxy platform, which is a free platform. And in purple, other free software that I used. So first step was the nuclear assembly of the DNA. Then we perform the mitochondrial DNA assembly and annotation. And finally, the annotation of the nuclear genome. So the first step, the nuclear assembly. We perform the assembly on reads, which are processed with the FastP in order to filter the low quality reads. Then with shovel, we perform the assembly, and with Quast and Busco, we evaluate quality and completeness. In the table, I reported the results of the assembly, and I want to focus the attention only on two numbers. The first one is the genome size. The genome size of our two strains isolated from humans result to be slightly bigger than the average that is represented by the blue line in the graph on the left. The second number I want to focus, the attention is the completeness that is measured using Busco. And as we can see from the table on the right, the completeness, the percentage of the completeness is comparable for our strain compared to all the available strain of this species. Moving on the mitochondrial DNA assembly, we use the same processed reads with FastP, and the tool for the assembly of the mitochondrial DNA was novoplasty. In this case, we need also a seed sequence that then is amplified by the software and create the circular sequence of the mitochondrial DNA. And we use a small portion of a gene of the mitochondrial DNA of the reference, the 7600. And then we use mFannot in order to annotate the mitochondrial DNA. The results is reported in the picture. And as you can see, is a single circular sequence that is around 53,000 base pair long. And it contains the 15 genes and 27 tRNAs, and then it presents 3 open reading frame and 4 endonucleases. Last step was the annotation. The annotation starts from the assembly obtained with shovel, and we use Augustus tool on Galaxy. For the Augustus tool and the annotation, we use the annotation of 7600 as a reference, and we produce the ab initio annotation. Then on Omics Box, with several tools, we perform the functional annotation. This annotation will then completed using anti-smash for the secondary metabolites and the factor P for the effectors. In the table, I reported the results. We found around 14,000 of genes for both strains and more than 40 clusters of secondary metabolites in both cases. Once obtained the genome, what we did is a comparison in order to try to correlate the origin and the biological result with the genomic information. First one is a genome alignment with Fastani. Then we build a phylogenetic tree using IQ3, and then we align also the CYP51A gene, which is one of the target of the azoles. First one was the Fastani alignment. Fastani produced a matrix which reports the percentage of identity for the aligned part of the genome. The results did not highlight any correlation between the isolation host and the similarity. But in this table, it's possible to appreciate the formation of two groups that are highlighted by the green boxes. Moving on the phylogenetic tree, we perform this phylogenetic tree based on 18 conserved genes. And also in this case, human-derived strains grouped with agricultural one, showing no host effect on phylogeny. Interestingly, also in this case, it's possible to appreciate the same two groups that were highlighted in the Fastani analysis. The alignment of the CYP51A gene. We tried this alignment in order to explain the different azole resistance that we found in the biological test. We aligned these genes and we aligned the two sequences of the two human strains and the sequence of the reference. What we obtained is that the two human strains were perfect identical, while in the case of the agricultural one, we found 3 substitutions that cause difference in charge. This difference is involved in the folding of the protein that has been demonstrated in Mycosphaerella that affect the sensitivity to the azoles. For this reason, the change in charge causes different protein folding, and this could be a starting point to answer and correlate the biological behavior, in this case, the sensitivity to the azoles, with the genomic information that can exploit it. Final step. So we found that there were no host effect on phylogeny. So can we exploit genomic information to identify possible determinants of virulence in human host? First, to answer to this question, we first search for the common genes shared by the two human strains, finding more than 13,000 of genes. Then we compare these genes with the entire pool of the reference, finding 118 unique genes. Interestingly, our effector workflow, coupled with phi-based functional analysis, identify an overrepresentation of genes closely related to virulence in animal host, as reported in the graph below. After this, given the availability of other Fusarium verticillioides genomes in databases, we further search for unique genes present only in the two human strains, and we found 6 unique genes that only the 2 strains isolated from humans had. So we start with the questions, with a question, and we close with a question. Are these unique genes involved in the pathogen-host interaction? To conclude, our research demonstrates the plasticity of the genome of Fusarium verticillioides, allowing them to infect both plant and animals. We are at the beginning to understand the genetic factors that contribute to clinical infection. These will help the future research and treatment strategies. Moving forward, we plan to perform RNA-seq analysis to further study the expression of these unique genes, and also we aim to create mutants of these genes to better understand their role in fitness and virulence. Thanks everybody for the attention.
Luca, thank you. I just also want to point out to everybody that Luca is one of the recipients of GXTX Foundation Travel Award this year. It's a foundation which was established in memory of James Taylor.
Thank you, Anton.
And we
Thank you.
Anyway, I'm just trying to see if there are any questions. I don't see any. Your genome sequences, are they contributed to the databases already, or what's the status?
Oh well, yes. This presentation is prepared by using data that were around April, like that. Then I move on Muse and prepare stuff like and study the Fusarium Muse. But coming back on the Fusarium verticillioides right now, I saw that in the databases are present already. They submitted in this time like 40 new genomes. So now I'm analyzing again the genomes, but yeah, I've submitted them and they are I'm waiting to publish it with the paper that I'm writing about this. But now I'm remake the comparison because I found 40 new genomes that were submitted in June, something like this. So it's something like I don't know the 6 genes. I think I don't know if they're still unique or not, for example. So these new genomes make something new in my mind to do.
I'm asking this because the state of genomes for these species is actually very bleak. There are lots of genomes at NCBI, but they're all of pretty bad quality. So I think
Yeah,
I think you can contribute significantly here.
yeah. If I have 30 seconds, if you look for Fusarium Muse, I did the chromosome level for it. So now maybe I work on only on the assembly in order to make it better and try to make the chromosome level. But the 7600 is very cool, let's say, with RNA-seq data, with everything. It's like 12 years that they are working on this genome and re-annotate and make new experiments. So that one is, I think it will be the reference for the species for all the time.
Thank you.
Thank you, Anton. Thank you, everybody.
Okay, again, this is Genomic Technologies. We are on our 5th talk of the evening. The next talk is by Mirian Tsuchiya from National Center for Biotechnology Information from NCBI. I apologize if I did not pronounce your last name correctly. And this is going to be about NCBI datasets, current status and future developments on the genomic data delivery at NCBI. I just want to emphasize that NCBI datasets is a critical and rapidly evolving resource, which is extremely useful for all of us.
Hi, my name is Mirian Tsuchiya, and I'm a bioinformatics data wrangler with the datasets team at the National Center for Biotechnology Information. In this talk, I will briefly introduce NCBI datasets, present some recent updates from the past 2 years, and talk about future directions on data delivery at NCBI. The NCBI datasets team comprises subject matter experts, developers, UI/UX designers, and product managers. We work under an agile framework, which means that we are constantly improving our products based on feedback from users, both internal and external. And everybody listed here, they're all co-authors on this presentation. So before I start talking about the past 2 years, let me introduce you what is NCBI datasets. NCBI datasets is a resource that simplifies the discovery, exploration, and downloading of biological sequences and metadata from NCBI. Datasets provides new interfaces for data access, including web interface, command line tools, and Open API. We recently published a paper on scientific data where we described in more detail our data packages, our guiding principles, and more information about everything we do. When determining the next developments, datasets has 5 main guiding principles that we follow. We adhere to FAIR principles, making sure the data is findable, accessible, interoperable, and reusable. We also want users to easily find the information they are looking for without the need of a profound knowledge of the ins and outs of NCBI. That includes making our tools easy to use for researchers at any level of expertise. We deliver data as data packages, which bundle metadata and data, and I'm going to talk a little bit more about those. And finally, we work hard to make our documentation as transparent and easy to use as possible, so users can find information on our data schemas, tutorials, and any relevant information. So one of the most unique principles that guide our development is the concept of data packages. A data package is a combination of data and metadata that can be customized according to the user needs. But the metadata is always included, and that's key to a data package. The main advantage of the data package is that it reduces the need to retrieve data from multiple databases or to know exactly where to find everything on FTP. This timeline shows some of the most significant events that happened since the Datasets project started in 2020. In NCBI years, Datasets is a relatively young project, and a lot has been accomplished in these past years. Between the beginning of the project in 2020 and mid-2022, we were in our beta phase. We didn't have a lot of releases, but we had a lot of breaking changes. Since 2022, we have reached a point where we have constant updates with well-documented version changes, but our goal is to keep it as stable as possible for our users. And for this presentation, I will focus on the developments of the past 2 years. So first, let's talk about some web updates. The first and the biggest one is the new genome pages. Since June this year, the old genome and old assembly pages were replaced by datasets pages. Merging these resources has been a long-standing desire for NCBI, and we know that we had users that would look for genomic sequences and metadata, starting either from genome and others starting from assembly. And depending how they searched, they would get different results and experiences. They now have a single entry point that supports the most common search terms. This transition was done in stages. For the past 2 years, we had placed information banners on the old assembly and genome record pages, informing the users that these pages would be redirected and after that retired. We understand that change is hard and messy, but now we have a better experience for our users and a more robust system of data delivery for NCBI. Another significant change is related to orthologs. Homologene was first redirected to datasets gene until its retirement earlier this year. And now we have access to invertebrate orthologs on the web. Through the datasets command line tool, users could already access those ortholog sets. Now both vertebrate and invertebrate orthologs can be retrieved on the web. Here I can show you how that works. From the datasets gene table, users can click on the ortholog set link on the orthologs page. And this link will take them to the orthologs page, where they can see all the species belonging to that set. If you're interested on more information about orthologs at NCBI, I recommend you to check out this talk by my colleague Dong-Ha Oh in the Binomes and Betnomes session. Another major change that affected all interfaces was the move from our version 1 API, which is parallel with the version 14 and up on the command line tool. I'll explain that. So on this timeline, I told you about our beta phase. Our beta phase is the equivalent to the first version of our API and our command line tool versions 13 and below. We are currently on the version 2 alpha of our API, and our command line tool is on version 16. All the changes I'm going to present, they happened starting on version 14. The first big change was the reorganization of our syntax. So here we can show that the ortholog menu was moved under gene, which makes sense. Sometimes we make changes based on technical decisions with the goal of making things simpler and easier to understand for the users. This move makes sense because we're talking about genes that are part of ortholog groups that were identified by NCBI curators. So orthologs, instead of being their own thing, they're under gene where they belong. Another breaking change with this version was the content of the data packages. Here I'm showing the contents of a genome data package as it was until mid-2022. We used to include all of those files, and the users had the option to opt out from them. But if you're using a program for the first time, you might not be aware of that. And the consequence is that you end up with a download size much larger and slower due to the data volume. The current genome data package have only the genomic FASTA and 2 reports, the catalog and the data report, which is the main metadata report. And instead of opting out of files, now users can opt in. It's a more deliberate choice to download all of those files. And what we observe is that users tend to download the default package, even after the change. Along with the data package content change, we also updated the flags. Instead of 1 flag per file, we have a single flag for all of them. And we are constantly adding new flags to make it easier to filter out data. Another big change with this new version was a better integration of data sets and data format. Data format is a companion tool to convert JSON line reports to TSV and Excel. Up to the version 13, users had 2 options. One, if they wanted to use data format, they had to download the data package and then use data format to convert the report from JSON lines to TSV. Or they could retrieve the metadata report using the summary command, which doesn't download, but it prints to the screen, and use a program like JQ to convert the information they see printed on the screen from JSON lines to TSV. The issue here is that you need to have a deep understanding of the JSON structure, and you need to learn JQ, and that's kind of a steep curve. Since the version 14, users can retrieve the metadata using the summary command and use data format to convert everything on the screen. And all it takes is a quick look at our documentation so they know which fields they're interested. One very important thing I want to point out is our version 1 API will be retired in the end of the year. So if you're still using that, please move to the version 2. And we're moving then from version 2 alpha to version 2. Another major change was the new taxonomy service. The NCBI taxonomy page is being replaced by 3 datasets pages. Those pages, new pages, are compliant with government accessibility rules, and they also contain all the information from the legacy taxonomy page and more functionality. On the main taxonomy page, users can find basic taxonomic information, links to the names page, and the taxonomy browser. And on the names page, users can find detailed information about nomenclature, type material, publications, nomenclature authorities, and download a report with that information. The report can also include taxonomic information for children and parent nodes. The taxonomy browser shows hierarchical taxonomic information about the taxon requested. In the main page, you also find links to genomic resources, such as the genome table, reference genome download, genes annotated, BLAST, genome data viewer. And at the bottom, you have links to other NCBI databases, including a section on type material. On the command line tool, the most visible change is the inclusion of a taxonomy endpoint to retrieve taxonomy reports programmatically. In a less obvious way, we also improved how the CLI handles ambiguous taxonomic names. In this example, we show what happens when somebody requests genome information for Drosophila, which can be a genus, a subgenus, a fly, or a genus of fungi. So where are we going from here? As I have mentioned multiple times, we are under constant development and change. We're always reevaluating our tools and incorporating as much feedback as possible. For the near future, we will continue to improve the taxonomy interfaces on the web, and our next big adventure is creating a more integrated experience for users looking for gene information. Datasets is here to stay. We provide users with a clear, well-defined path to find and download data across different interfaces at NCBI. We understand that this is a big change from the older pages to the new ones, and that change can be very uncomfortable and sometimes scary. But you're not alone. Our team and NCBI as a whole embrace and welcome feedback, and we do as much as we can to support our users. So please feel free to contact us. You can use the help desk email, you can find us on GitHub, or you can use the feedback button the yellow feedback button on our pages. If you include your email, we can get back to you and respond to your concern. I would like to thank the datasets team here and all other teams that work with us at NCBI and our users. Thank you so much.
Mirian, thank you very much. Something happened with the question board. I guess it got reset, so it's absolutely white, but that's okay. I have no shortage of questions. So this is, of course, biodiversity conference, and sometimes when we think about biodiversity, we think about big things that we can see. But one of the critical components of NCBI datasets, and I'm not sure I understand hierarchy completely, but of course, NCBI viruses is also part of you, right? So
Correct. Correct.
And one of the challenges there is that, for example, if you look at things like, avian influenza, COVID, or anything else, there is no single reference. And especially with viruses with high divergence, such as avian influenza, you have a lot of, it's very hard to pick what the reference genome is. So are there any thoughts of organizing this somehow? I'm not saying you should use genome graphs, because I don't think very few people know how to do that at this point. But is there a way to, if you, for example, want to survey variants of influenza in some country, to pick a proper reference using tools like Vapor or some K-mer-based, essentially choosing reference based on your input data? Do you have any ideas there, or should I ask some?
I think that's a better question for the virus team.
Okay.
Datasets is on the front end of delivering data and not really curating any of the data that's delivered. So that would be a good question for the virus team. But yes, they're constantly working to improve references and get data as fast as possible, especially in times when we need that data there.
The moment I started complaining, questions appeared. So from Katrina Hauff, thank you for making NCBI datasets. I have used the command command line tool for data retrieval and bulk download. It's a game changer. Will you in the future directly integrate SRA for transcriptome entries for species and strains? I currently query SRA separately.
Yeah, we have been getting a lot of requests to have SRA data delivered through datasets, and currently we don't do that, but we have been the SRA team and datasets team have been talking about plans to make that data integration easier for users. We don't have a timeline. I don't have a clear answer as this is happening when, but we hear you. Please keep sending that feedback. That helps us really keep the conversation moving on that direction.
Thank you. Another question from Eric Jarvis. Thank you for the changes that your NCBI team has made. There are some changes I like and some other ones that I preferred. Are you getting feedback to the community before making changes, and who is the appropriate person to contact?
Oh, absolutely. So if you send any feedback to the feedback button or any email, GitHub, anything, if you say that's specific regarding a genome page, an interface or something, that will get to us. So all of those channels are a good way to reach us. And in terms of who we talk to, we constantly have user interviews before any of those major changes happen. We create mock-ups of our web pages, put that in front of users, see if they can find the information we believe they should be able to find, gather this feedback, and then that's how we keep evolving. Obviously, that we do that first round before we release a page, and when we make new changes, we do another round of those. But we are constantly taking feedback from the community. So please, we love feedback. It's real. We read everything, and we really appreciate any input you can have on our pages.
Thank you. Yeah, in my experience, you really like feedback, and thank you for that. I think it was a wonderful collaboration with you.
Absolutely.
Well, thank you. So we are continuing. For those of you who are just joining, this is again Genomics Technologies session, and we have the last talk by Uja Strop. And this is on high quality eukaryotic genome annotation with NCBI public EGAP-X pipeline. So this is the second talk in this session on genome annotation. I would say third talk on genome annotation. Because without this, genomes are just big text files. Please.
Hi, I'm Pooja Stroh. I am a bioinformatics data wrangler in the RefSeq team at NCBI. Today, I'm going to present to you a high-quality eukaryotic genome annotation pipeline called EGAP-X, developed by our team. At NCBI, we have seen a continuous growth of genome submissions every year. Not only is the total number of genomes submitted increase each year, but also the number of distinct and new species. Currently, there are about 36,000 genomes from around 15,000 species. But a big problem is that the vast majority of these genomes are unannotated. Only about 19% of the genomes are annotated by users. Unannotated genomes are not that useful, so we want to fill that gap and allow users to be able to submit more genomes that are annotated. So what do I mean by annotation? I'm referring to adding information about the genes and proteins made by the genome. That includes both the structural annotation, identifying the complete structures of the genes, transcripts, and proteins, and functional annotation like gene and protein names and what they're related to through orthology. All of this provides information to help understand the biology of the organism. We'll compare it to genes and proteins in other organisms, and from an NIH perspective, apply this information to improving human health through understanding disease and drug discovery. With the great expansion of genomic data now being produced from many eukaryotes, NIH recognized a need to improve resources to support comparative genomics and funded what we call the comparative genomic resource. This includes development work at NCBI on new and improved tools, including a pipeline for eukaryotic genome annotation. EGAP-X stands for Eukaryotic Genome Annotation Pipeline External. The external X part means that it is public. The main goal of EGAP-X is to be able to produce submission-ready annotation for GenBank. Out of the box. The biggest barrier of annotation tools has been that they don't always produce error-free annotation that meets GenBank standards. We want to be able to remove this bottleneck and streamline annotation submission so that it is available to the community. Our goal is that the users can download and run it themselves easily. It should be very easy to configure. It should run in multiple environments, whether it is in the cloud or in local cluster. The workflow is created using Nextflow and it runs a Docker container. Multiple jobs are parallelized depending on resource availability. This EGAP-X pipeline is largely based on the RefSeq EGAP pipeline, which was internal to NCBI and not public. And EGAP-X has been under continuous development for almost 20 years. In EGAP-X, we're restructuring, optimizing steps, largely aiming to carry over the functionality that we have used for annotating genomes from over 1,000 species for RefSeq. Now let's go over the EGAP-X pipeline. First, you provide a config file, which has the information about the genome you want to annotate, the tax ID, and RNA-seq data. This information flows into the RNA-seq alignment task, shown here in red. We require RNA-seq data. From years of experience, we have seen that RNA-seq makes a huge difference in annotation quality. RNA-seq reads are aligned to the genomes using STAR, Then we analyze which runs were sequenced using stranded technology and use this information to help resolve closely packed or overlapping genes. Then the alignments with the same splice structure and similar start and end points are collapsed to reduce the volume of the alignments. And we use a local abundance filtering process to weed out noise and rare splicing or retained intron events that are sometimes referred to as transcriptional noise. Next, for the protein alignments, EGAPX automatically chooses the protein set. We have automated the configurations as much as possible to keep it easy for the users. EGAPX aligns a taxonomically related protein set against the genome using miniprop. Alignments are then ranked and filtered according to quality. We also use RNA-seq data to help filter the protein alignments. It helps clean up the ambiguities that show up in protein alignments. The RefSeq EGAP pipeline has also used long-read transcripts alignments for almost a decade. And this logic will be adding to EGAPX this fall, and this tasks are shown here in green. The yellow boxes is the part where everything comes together. This task, NOMON, takes the evidence from the protein and RNA-seq alignments. It combines them into chains that are optimized to represent whole transcripts and proteins. NOMON will also supplement these models with ab initio predictions where needed. Ab initio isn't used much. The majority of the models are based on the alignment themselves, but ab initio can help to compensate for gaps in the evidence. EGAPX automatically optimizes ab initio parameters for new species. It starts with existing parameters from the closest available species and then automatically iterates to optimize the hidden mark models. We will be adding a task for tRNA, ribosomal RNA, and other short RNA predictions in the future. So at this point, we have the structural annotation, including both protein coding genes and link RNAs. To provide functional annotation, EGAPX categorizes gene by biotype, such as protein coding and pseudogenes, and identifies orthologs. Orthology is computed using protein alignments to reference protein and refined using local synteny. We use the orthology along with additional alignments to Swiss-prot proteins to provide functional information and name genes and proteins. Then all of this information is packaged in a format that is suitable for GenBank. We want to make EGAPX easy and highly automated. We provide pre-computed sets of evidence protein chosen for specific groups of organisms. These are built from RefSeq proteins from well-annotated genomes that are representing the diversity in the group. EGAPX includes logic to automatically pick the most appropriate set. Within the vertebrates, we include specific sets for mammals, reptiles and birds, and ray fin fishes. We are also adding the general vertebrate set to include the outliers such as sharks. Similarly, in the arthropod set, we have 6 sets for different clades and then a general arthropod set. For plants, there is 1 set for monocot and 4 sets for eudicots. If you are working with a metazoa or flowering plants and you have something that is not covered, please talk to us. Right now, there are additional work we need to do before we can support fungi, protists, nematodes, and green algae. Now let's look at what do you need to get started with EGAPX. So the different parts of EGAPX, you can get it from our GitHub repo, ncbi/EGAPX. Currently, it is in alpha version. From there, you can find a runner script, the next full pipeline. The Docker image will be pulled by the runner script automatically. There is documentation and several example files for you to try. On the computational side, you will need a cloud platform such as an AWS instance or AWS batch. You can also run it in local cluster that's available to you. You will need at least a node with 32 CPUs and 200 GB RAM at minimum. In the future, we're hoping that Galaxy will be able to support eGAPX. The disk space that you need is going to be is huge depending upon how much SRA data you're going to use, but at least a couple or a few terabytes of disk space is needed. So again, we want to make running eGAPX as easy as possible. You do not need to provide too much data. We have automated steps to select protein set, ab initio parameters, and orthology data sets. You only need the genome you want to annotate, the tax ID, and RNA-seq data. You would put this in a YAML file. Reads can come from NCBI SRA or from local paths. You will find examples of input files in the documentation. To run eGAPX, you would run the Python script that you downloaded, give the input YAML file, provide the executor that defines your cluster resources, the output directory, and a work directory. The final output will be in the output directory, and work directory will consist of all the intermediate files. We provide executor configs to get you started. You may have to edit these in accordance with your cluster specifics. Our main motivation is to make it easy for you to run, provide consistency and scalability. Considering the big genome projects like the Earth Biogenome Project and similar large-scale projects, we want to make sure eGAPX can scale up. The next flow pipeline provides you with some graphical reports of your eGAPX run with details about resource usage. These are in HTML format in the output directory. Here you can see the CPU usage of star and miniprot. They are the highest here. In the bottom, you can see multiple miniprot jobs and star jobs and how long it took to run them. In this case, few minutes. This is a great way to monitor the resource usage for your runs. So we've been working to evaluate how well eGAPX is performing and make further refinements. One challenge is that the RefSeq eGAP annotations are one of the most commonly used benchmarks, and eGAPX is an updated version of eGAP. So one strategy we're using is to benchmark against the Drosophila melanogaster annotation from Flybase in comparison to other tools. This is focusing on just the protein coding genes, and we compared it to Breaker and GeneMark ETP. You can see eGAPX has picked over 85% of the exons and over 80% of the genes as compared to what's found in the Flybase annotation. The transcript annotations are lower at around 60%. Flybase is extensively manually annotated, including rare transcripts, which makes it harder for prediction methods to find all the transcripts compared to just gene-level comparisons. High precision values mean lower noise in the annotation or features not annotated by Flybase. We see over 90% precision for exon level and gene level annotations and a bit lower for transcripts, which we're still working to optimize. In the future, we will work on boosting these numbers and do comparison using other organisms. We compared the annotated protein to the Busco diptera set. Busco comparison allows us to assess the completeness of the assembly, and in this case, we're assessing the completeness of the annotation. As you can see, eGAPX has annotated 99.4% of the proteins in the diptera set. This number is quite close to the assessment of the Flybase annotation, and a couple of percentages better than Breaker and GeneMark. For EGAP-X, we are putting a big emphasis on iterative development. While we can test the software in our local environment and in AWS, we cannot test it in the plethora of HPC environments that are out there. So we took the approach of releasing a partially functional alpha product early to start getting feedback and optimize where the pain points are while we continue to add features. We started off targeting specific users for feedback, but what has been most valuable is our users in GitHub, many of which we don't know who they are, but their feedback has been very informative in the progress of EGAP-X. Most of our feedback comes through issues in GitHub, which we use to identify bugs, pain points, and prioritize what to work on improving. Our developers then refine the pipeline and test it thoroughly before making another release. So you are all welcome to try EGAP-X if you have not already. Finally, I want to thank the EGAP-X team for working diligently in producing this product, and also to Terence Murphy, who is the lead for EGAP-X and CGR. This work is done as part of the NIH comparative genomics research, which is working to maximize the impact of eukaryotic research organisms. Thank you.
Thank you for this presentation. We have some questions. How does the accuracy of EGAP-X compare to that of the RIF-seq EGAP? This is a question from Maria Stanke.
Hi, thanks for the question. We have been comparing EGAP-X and EGAP results. They are really close, but not the same. And that's one of the things we're trying to do is get as close as we can. There are some differences in like the protein sets we're using, some of the algorithms that are used within it. So right now, I think that it's like over 95% identical results we're seeing. So we're working on trying to find the differences, what's causing them, and how to resolve them.
Thanks. I just wanted to clarify. So EGAP-X is already on Galaxy, so I put the link so you can run this right now. I think at least some users in VGL are already doing this. So we do need more people to run it to give us feedback. Just so they give us feedback, then we give you feedback. This kind of how this works. I'm just looking if there are any other questions. No. Yes, in terms of resource allocation, so I mean, one of the issues, of course, is that this is a big thing with many pieces, and these pieces have various requirements. Obviously, if you run star, you need a lot of memory because this is suffix tree-based thing. How do you have any? Because it kind of creates problems for external users, because if you want to use your resources in an optimal way, it's not easy from a single, if you run this workflow as a black box. Do you have any ideas for fine-tuning, specifying resources for specific tools within that workflow? Or I guess maybe I'm not sure.
Yeah, we do not have any like automated way of figuring out ahead of time how much you need. Right now, we're still in the development phase that these numbers are preset in our configuration. But as we progress and as we identify, with more runs and results, we will have a mechanism to know ahead of time how much, so that we're not wasting resources and we're trying to be as close as what's needed for that run.
Yeah, so that would be because that's one of the problems for us, is that we essentially, the way we run it is a black box. So we provide it with fattiest node we have, because we need to ensure that it runs, that the star runs efficiently. But most of the time, it just wastes resources on that node because it occupies and uses very few threads or not that much RAM. So, but anyway, I must say that we are, at least we are in consistent communication with you, so we can probably solve these things in the future. Any other questions? I don't see any other questions on our question board. I have a lot of technical questions, but I don't think I should be asking them here. I'll ask them offline. Well, I think that's it then. So I just want to thank all the speakers for agreeing and recording very nice videos and being here to answer questions.